Hello, welcome to Pharmaceutical Science 505 Bioinformatics. In today's video, we're going to talk about some basic terminology in bioinformatics. In order to learn bioinformatics, to start somewhere, we need to know some basic terminology so we can ask the right question. Either that's online asking Google or Google search or asking ChatGPT. You need to actually ask the right question in order to actually get the answer that you wanted, right? So let's start with some basics. So the first thing is this figure. So here is how DNA produces mRNA, and mRNA can be translated into protein. Let's take a look at this figure. So the first part is we have a promoter. A promoter is the short sequence on nucleotides on DNA that start the transcription of RNA by RNA polymerase. In other words, promoter is the starting region of the RNA coding sequence. Here, we see this is called the RNA coding sequence. Why it's called the coding sequence? Because in RNA, there are different regions. There are actually coding region and the regulatory region. The coding region produce mRNA, and mRNA eventually produce protein. In other words, the RNA coding sequence are related to the final protein produced. The regulatory sequence are not known. Many of these are believed to not result in protein, and some of these are called non-coding RNA. However, in recent years, more and more of these non-coding RNA has been shown to actually produce some of the protein. The coding region is the portion of genome that's translated into RNA, which in turn codes protein. And that region is also called exon. The regulatory region is a segment of DNA that controls whether or not, to what degree, a gene will be expressed. In other words, these regions does not produce protein, however, they control protein production. From here, then we can see there is the exon and intron. So exon is the coding region that actually produces the protein, and intron is the one that get deleted. So here there's an important concept called alternative splicing. So alternative splicing is a regulated process that during gene expression allows a single gene to code for multiple proteins. For example, in this example, we have 3 exons and 2 introns. And then we have a head and a tail. The 3 exons can be expressed together. However, it can also go through exon skipping, meaning that we can skip one of the exons. Say we do not express the second exon, only express the first and third exon, and that will be a new protein. However, this new protein shares the same first and third part of the original protein with 3 exons expressed. There's also the mutually exclusive exon. Mutually exclusive exon means that one of the exons is going to be expressed but not together, meaning that this piece of RNA is designed that this exon can be expressed, and then one of the other two, either the second one or the third, can be expressed one at a time. So it can be either 1, 2 or 1, 3. There's no such thing as 1, 2, 3, and that's called mutually exclusive exon. In alternative splicing, there's also the alternative 5 splicing site and alternative 3 splice site. The choice of different 5' splice site in the pre-mRNA changes which portion of the RNA are considered exon and intron. In other words, here exon and intron, they could be interchangeable, depends on how you actually start 5' splice site. Similarly, there is also the choice of the 3' splice site, which changes the exon-intron structure of the mRNA. This will further complicate the alternative splicing. There's also things called intron retention, means that the intron is retained in the spliced mRNA, so unlike the typical splicing where introns are removed. However, remember that mRNA is the one that's producing protein. So if an intron is actually retained in the mRNA, that means the intron will be expressed. Next, we're going to talk about three important concepts. One is reading frame, second one frame shift, and third one is single nucleotide polymorphism. So, what is a reading frame? So a reading frame is basically where the protein start and where the protein end. So the reading frame start with a start codon and stop with a stop codon. And in between is the open reading frame. A frame shift is basically a genetic mutation which shifts the reading frame used to translate mRNA. Because the codon has 3 nucleotides, the frame shift has the +1, +2, +3, -1, -2, and -3. So +3 is typically just skipping the first amino acid because the sequence essentially is unchanged except it's skipping one of the amino acid. But +1 and +2 means that it shifts 1 nucleotide or 2 nucleotides and change the whole meaning of the protein. So the protein will be expressed in a totally different way. The single nucleotide polymorphism means that there's a single nucleotide that's changed. Typically, that only results in the change of 1 amino acid. And SNP could be fatal if it's changed in the binding pocket or the active region of an enzyme. Next, we're going to talk about sequencing. Reads are sequence of DNA that has been generated by sequencers from fragmented DNA samples. So in the typical sequencer, we have the short reads and the long reads. The very popular Illumina sequencer are short read sequencers, typically 150 base pair or 250 base pair long. Long reads, on the other hand, can read hundreds of thousands of base pair at a time. Short reads has the advantage of a higher throughput since they actually shear the DNA into smaller pieces, and this process can be greatly accelerated because the short reads are more unified. The long reads, because the read length actually varies a lot, creates lots of variability, and that's why the long read is actually less stable and therefore lower throughput. However, when you try to assemble the read, here we see the short read, there could be sequence gaps, meaning that certain part of the sequence may not be actually covered by the short reads. That's the risk of having the short reads. This doesn't really exist for the long read because the long read long enough, so the gap is less likely to appear. Sequence alignment is the process of arranging these sequences from the sequencer to the reference database based on their region of similarity. Next, we're going to talk about the FASTA format. FASTA is a file format for representing nucleotide sequence or peptide sequences in which nucleotides or amino acids are represented using single letter codes. Here I'm showing you a protein FASTA. RNA or DNA sequence are similar to this. The first line is the title line. The title line gives information about this protein or gene. There are 3 parts of it and divided by the vertical line. The first part here is showing SP, meaning Swiss prot, means this data is getting from a database called Swiss prot. And then it has a UniProt ID number. This number is uniquely assigned to a protein. In this case, this is the SH3 binding protein 1. Then it gives the full name, the human readable full name, that's called human SH3 domain binding protein 1. Also gives the gene name of this protein, SH3BP1. The organism is from human. The PE here annotates the protein existence, and the SV annotates the sequence version. The second line and below are the sequence of this protein, here annotated by the different amino acid. And then the FASTA file just go on and on to the next gene, which start with a greater than sign again, and then that's another SP. The next thing we're going to introduce is BLAST. So BLAST is a basic local alignment search tool. It's a tool that can find regions of similarity between biological sequences. The program compares nucleotide or protein sequence to sequence database and calculates the statistical significance. NCBI has a BLAST tool online and you can use it for free. Here is the result generated by the BLAST. As you can see, these arrows point out to the difference in the two sequences. The first sequence above is the query, and then the sequence aligned is called the subject. The consensus sequence is the most commonly occurring amino acid or nucleotide at each position of an aligned series of protein or polynucleotides. Next, we're going to talk about gene ontology. Gene ontology provides a controlled vocabulary to describe gene and gene product attributes in any organism. People who study worm and people who study mammals may actually describing the same process by different words. And the Gene Ontology Consortium is trying to unify this by providing a controlled vocabulary. In short, it contains three different big categories. One is biological process, which, for example, can be the TCA cycle, glucose metabolism, DNA repair. Basically, is a thing that you study in biochemistry. Second is the location. It's called cellular component. It can be the cell membrane, cytoplasma, extracellular matrix, nucleus. The third one is the molecular function, which shows at the molecular level what the function of a molecule is. For example, the toll-like receptor binding means that this protein binds to the toll-like receptor. Adenylate cyclase activity means that this thing works as adenylate cyclase. Or oxygen transportation means this protein transports oxygen. Let's actually take a look at how it works. We have the Amigo provided by geneontology.org. The first thing we do is we select for organism. We select for the organism that we want to study. In this example, we're going to use human. And here are the three categories: biological process, cellular component, and molecular function. Let's expand them. Biological process, here show the biological process. And let's say biological process in the cellular process. And in the cellular process, there's all of these terms. And if we open cell cycle, here's the cell cycle process, the mitotic cell cycle process, and if you further open them further, there will be all these terms related to the cell cycle process. For example, the G0 to G1 transition, and then there's the negative regulation, positive regulation, or just regulation. We don't know if it's positive or negative. And these terms, when you actually click on them, it has an explanation, but also if you click on the term, it will lead you to all of the genes that actually that has this term, that's labeled by this term. And here are all the genes in human that's labeled by this term. There's total annotation 41, showing only the first 10 of them. So that's gene ontology. Gene ontology terms are assigned by human. There's an expert panel that reviews these regularly. Okay, so much for today's video, and I'll see you next time.